This is an A-level biology presentation looking at Sanger sequencing in terms of genome projects. To get started, there's a number of terminologies that you should be familiar with. Go through each of the ones on screen, come up with a definition, and when you're ready, you can go through and check your answers. Pause the presentation now, and when you're ready, restart to check your answers. Well, totipotent is in reference to stem cells, and it's the most diverse stem cell you can get. You've got specialized cells, which are cells that have had certain genes turned on and off to give them specific functions. We have differentiation, which is the process in which a stem cell develops into a specialized cell. The gene pool refers to all alleles in any population at a specific time. The genome is all the genetic material in an organism. Isolation, remember we talk about reproductively separated, and proteome is all the proteins produced by the genes of an organism. Remember there are 4 types of stem cells, the extension question. If you haven't had a chance to have a go at it yet, pause the presentation and try and remember the 4 types of stem cells as well as any characteristics of each type of stem cell. Pause now. The 4 types of stem cells are totipotent, pluripotent, multipotent, and unipotent. Remember, the ones that come from the zygote, from the embryo, are the totipotent, and then as we progress through, we can get them from things like the placenta all the way through to adult stem cells. But of course, the more mature the stem cell, the less differentiated it can become. The Human Genome Project. The Human Genome Project looked at 3 billion base pairs, 20,000 genes. It took well over 10 years to put together, and the sequences are actually available online. You do have to pay for them, but they are actually available. The question is, why did we bother? It was huge amounts of money involved. Why did we bother? Spend a minute, no more than a minute, come up with some ideas over why we bothered to actually determine the human genome. When you're ready, you can restart the presentation. Well, there are lots of reasons why the Human Genome Project was important. We have a large number of diseases that are not infectious. They do not spread from person to person. Looking at things like cancers, Alzheimer's, motor neuron disease, Parkinson's. These are all specific to individual people, and they're all determined partially on the genetic makeup of the individual. And by knowing the genetic makeup associated with each of these different diseases, we can actually start treating these diseases and even eradicating these diseases by correcting the faulty parts of the human genome. But we can only do that once we actually know which of the genes are actually involved with which disease and the sequences for the genes, because remember, there's a lot of different combinations of the DNA sequences that will give you the exact same amino acid sequence because of the whole concept of the generic code. So how do we actually determine the sequence of the human genome? Well, what they've looked at is an entire field of research is dedicated to purely determining, firstly, the sequence of DNA bases inside any particular organism, but then going much further by determining what each of those bases actually contributes to in terms of coding, non-coding, and in terms of genes, characteristics, and alleles. There's a huge amount of work being done to cover all the organisms, and we've barely begun to scratch the surface of even the human genome. We still don't know what large sections of DNA code for. We do have this thing called a shotgun sequencing, which is where you break the sequences of DNA up into small sections, and then you basically put it together like a jigsaw puzzle, looking at computer patterns and stuff. It's a hugely time consuming, even with computers, it's hugely time consuming. And we're going to look at one particular method that was used to determine, or can be used to determine, the sequence of bases in any particular section of DNA. Now remember, proteome, when we talk about proteomes, we're talking about the proteins coded for by genes. Now you can be asked a question making comparisons of investigating proteomes in simple organisms and investigating the proteome of complex organisms. And the most important point to remember is the fact that in simple organisms, often we refer to things like prokaryotic, but it could be eukaryotic as well. If it's prokaryotic, remember we have no introns. We have the exons, which are the coding, but there are no introns. Hence, in prokaryotic organisms, we don't have splicing as part of transcription. If we have got introns, then what we have there is when we sequence the entire DNA sequence, we've got a problem of which bits are coding and which bits are non-coding. Even going further than that, you have to determine of the non-coding bits, what are the functions of the non-coding bits? Are they there because they're just junk DNA and don't serve a function, or is there something more complex going on that you need to be able to determine, and how do you determine that? There's also a further problem in terms of proteomes. Even if you know the protein polypeptide sequence, even if you know every amino acid in that protein, because of the degenerate code, you have the issue that you can have different combinations of DNA giving you exactly the same sequences. So it's even more complicated, which is why when they're sequencing DNA, they don't take just one organism, they take large numbers of organisms to look for patterns overall. So one of the ways we can determine a DNA sequence is using what's called a Sanger sequencing approach or technique. You've got the information in front of you. You start with a single-stranded DNA, some primer, some DNA polymerase, and then what we refer to as D nucleotides. So you've got ATP, TTP, CTP, and GTP with the letter D in the front. They are other alternate versions of adenine, thymine, cytosine, and guanine. And you also have a version called DD-ATP, DD-TTP, DD-CTP, and DD-GTP. So there's lots of D's knocking about in this one, but there is a difference between the D version and the DD version, which if you look at pictures 3, you may be able to work out. Pause the presentation, spend no more than 5 minutes trying to determine what is going on in this process. And when you're ready, restart the presentation and we'll walk you through it. So what we've got is a single-stranded DNA, some primer, some DNA polymerase. Well, if we've got single-stranded DNA, primer, and DNA polymerase, we have all the things there for DNA replication, so we can make lots and lots of this DNA sequence. We can replicate it as many times as we want. What we then have is what's called the DD versions. And the DD versions, if you look at number 3, when there's a DD version, when you get to that particular nucleotide, it actually stops the process of replication. So what you end up with is that particular sequence in the top left of single-stranded DNA starts with a letter A. Well, if we go to the container where we've put ATP, TTP, CTP, and GTP in every single container. But what we add into each of the containers is only one of the double DNTPs. So in the red beaker, we added the A version. In the blue, we've added the T version. In the yellow is the C version. And in the pink is the G version of DD NTP. Remember, the N just refers to any of the 4 versions. But in all 4 containers, we have the single D version of all 4 nucleotides, A, T, C, and G. When we start to replicate DNA sequences, what we will find in the red container is that anywhere where the adenine is the base, where there's an A, there is a chance that a D-ATP will be inserted by the DNA polymerase, but there's also a possibility that the DD-ATP will be inserted. If the double D version has been inserted, then the sequence no longer replicates any further and stops immediately at that letter A. Hence, if you look in picture 3 at the very top, we always have an A on the very end, or we run out of material when we just do the entire sequence from start to finish. So in the first of the red samples, that first letter A is made up of DDATP. If, however, by pure random chance, the first letter A happened to be a DATP, then we continue on with the sequencing process until we get to the next letter A. And in the next letter A, if it is a DDATP or DDNTP, you will have the sequence stop. If, however, the second A is a DATP, the sequence continues. And exactly the same thing happens in all four versions, except that instead of stopping at A's, the second one in the blue container has DDTTP, so it will only stop on T's, and then sometimes it will continue and sometimes it will stop. For the yellow beaker, it's the C's, and for the pink one, it's the G's. And what we ended up with is when we actually spread these things out, because all of the sequences are different lengths, they will have different densities and therefore will spread out different distances along in gel electrophoresis. And we'll come back to gel electrophoresis another time. But when we spread them out, because they go in order with the lightest ones can go the furthest, and the heaviest, longest chains go the least amount of distance, we can determine the sequence of the particular original strand. Because in part 4, we've taken a sample from the red, the blue, the yellow, and the pink container. We've put them down in their own little lines, and we've done what's called a gel electrophoresis, which for now you can just imagine it as a type of chromatography. And depending on the length of the particular sequence, we have it go to different distances along the gel. So we can see in number 4 that at the very bottom we have an A, which represents the A in number 3, which is on its own. If we look through all of number 3, the next longest chain with 2 nucleotides is found under the blue container with the letter A and T, which is why in number 4, we look at the second furthest, we can see that it came from the DDT container. And we can work our way along going the 3 nucleotides will be the third furthest away from a starting point because it's the third shortest. And if we look to number 3, go all the way down to find the one with 3 nucleotides, it's in the pink container ATG. And hence, if we look on number 4, we can see that we have the G represented by the pink dot in column 4. Now the reason we use colors is because if we can attach different colors to adenine, thymine, cytosine, and guanine and the DD versions, what we can actually do is we can actually produce a colored sequence that can be interpreted directly by a computer. And if we can set this all up in the computer, it literally becomes automated. And it was one of the biggest advances in terms of determining genetic sequencing was the development of technology that automated the process of working out the bases. Originally, this all had to be done by hand with large black and white spreads of gel electrophoresis, and you literally had to look for all the little dark marks and work out where each of the bases occurred. Whereas now with the red, blue, yellow, and pink, it's very quick and easy for a computer to interpret the colors and literally spread out a list of here's the nucleotide sequence. Please do go back over this. It's a complicated little process, but once you rack your brain around it, it actually makes quite a bit of sense. And it's one of those ones in the exam that they quite are fond of in terms of interpretation and analysis questions. So do take the time to go through it. What I've done is give you a very condensed version of the previous description. I can't read through it. It may be easier to follow than the verbal description, but make sure you are aware of everything there and make sure it does make sense as to what's going on. What you can see on the right hand side is exactly the same as the version that I gave you previously, insofar as you've got your columns for A, T, G, and C. However, this time it's a black and white version and we simply have banding rather than colors. And you can see if you read this sequence, you can see at the bottom, just off the screen is a T, then an A, then a C, G, A, G, A, T, A, T, A, T, G, G, C, G, and you can literally go across and read the entire sequence of that particular DNA strand. Now that's really useful in terms of determining the sequence, but of course what it doesn't do is give us any information about what that sequence is actually responsible for. So to finish off, you have to talk continuously about the material in the lesson for 2 minutes. Remember, any repetition, a pause, or any mistakes, and you have to start your 2 minutes again. You can talk to a family member, a pet, a mirror, yourself, whatever you can get your hold of, it's not a problem. What you're looking for is, can you talk for 2 minutes continuously about proteomes and the risks and problems associated with looking at proteomes and determining DNA sequence, and of course Sanger sequencing.